Sorry, I know it just means that I couldn't really hear clearly. So to repeat your question, what you're asking is that did I look into other types of milk and okay, thank you for your question. So my answer with that is that, so I haven't actually had a chance to look into other products of milk type yet, but forward to my PhD, I will be looking into other types of food, not just milk in particular. So I'll just add on a little bit more. So bacteria, just like humans, they can change very easily. So if you feed them another medium, they will, in terms of the volatile profiles, it will be quite different. So for example, if let's say I were to feed them spaghetti, the volatile profiles will be quite different from what I'm seeing in milk. So in the end of the day, we will need to put in a lot of work in terms of like doing for the screening in terms of controls. However, if let's say we are able to get all these preliminary work done at first, and after that, it could just be smooth sailing.
Okay, great. Thank you so much.
Thank you very much.
Okay, sorry, minor technical difficulties. I couldn't find a skeleton on the screen. Okay, so we have our next speaker, Rushali Dargan, from the Université de Québec at Trois-Rivières. Okay, go ahead.
Thank you so much for the introduction. Today I'm going to be talking about the use of two-dimensional gas chromatography to understand the decomposition order, and this study was specific to Canadian environment. Just a brief introduction. So following death, the process of decomposition commences, during which all of the soft tissue is lost, and what we are left with is dry or skeletal remains. And decomposition can be viewed as progressing through several stages. Namely the fresh stage where the donor has recently arrived and it has a fresh appearance. The middle stage where the donor is bloated and there's a rapid loss of soft tissue, so active decomposition. And then the late stage where the donor is desiccated or largely skeletonized. The process of decomposition is actually governed by several intrinsic and extrinsic factors. And to name a few, there's temperature, rainfall, presence of scavengers, and soil conditions. And so because all of these factors depend on the geographical location, it becomes important to conduct all of these studies at different geographical locations. The reason we study decomposition order is because of its application to cadaver detection dog training and to understand their capability. Essentially, during decomposition, all of the biomacromolecules such as proteins, lipids, carbohydrates, and nucleic acids, they break down to simpler forms. And in their simplest form, they can exist as volatile organic compounds. And it is these volatile organic compounds that the cadaver detection dog actually identifies when it's trying to look for an event of decomposition. And since the cadaver detection dogs cannot be telling us which key compounds is it identifying to as decomposition related, we do have modern day instrumentation to help us with the same, which brings me to my methodology. The current study is conducted at a REST facility, which is the first outdoor taphonomic facility in Canada. And it's just an outdoor secure woodland environment where donors are decomposing naturally. And we were collecting VOCs from air matrix. So the first step was to actually accumulate the air above the decomposing remain, which was achieved by placing an aluminum hood that looks something like this above the donor. And then the air sample was collected onto the sorbent tube, which was then thermally desorbed and injected for analysis. And the instrument we used was GC-TOFMS. And the third part was the data processing, for which peak finding was conducted using the CHROMATOGRAPHY software. And then we had custom R scripts that was actually identified compounds that was significant to the sample itself, as opposed to the background or the control samples. The results, in this study, we studied 8 donors, 4 of which were decomposing at warmer ambient temperatures and 4 of which were decomposing at cooler ambient temperatures in the year 2020 and 2021. And key findings, we detected about 1,400 VOCs across all of our 8 donors, all of the stages, which is why that's a high number. And just to name a few, there's dimethyl trisulfide, dimethyl disulfide that was significant. And as mentioned yesterday, these are compounds that smell really badly. And I did take that image from Professor Waska's presentation. But in our lab, we actually love smelling because these are the compounds that actually give the characteristic odor of decomposition. And so they are compounds of interest to us. And because it's a really high number of compounds, we do classify them into their compound classes. And just based on the number of compounds that belong to each of these groups and their normalized areas, we found esters, nitrogen-containing compounds, and alcohols to be quite significant regardless of temperature, just generally. And ethers, aldehydes, and cyclic aliphatics to be less dominant. And when we look at stage dependency, we saw which is the bloat and the active. There's acids, aldehydes, esters, and nitrogen-containing VOCs that were again dominant. And during the late stages, there were linear aliphatic and cyclic aliphatic VOCs that were dominant. Again, we did some PCAs, and here is a PCA of all of our 8 donors. As you can see that the donors decomposing at cooler ambient temperatures were tightly clustered, while there was a great deal of variability along all of our principal components for those decomposing at warmer temperatures. And this was consistent with the fact that the donors decomposing at warmer temperatures progressed a lot rapidly, and we also did detect a lot more VOCs at warmer temperatures. And this is again consistent with what has been reported previously. And a PCA of all of the different stages. And so here is for the 4 donors that was decomposing at warmer temperatures. And there was subtle variability between the late and the middle stage owing to specific VOCs that were dominant in each of these stages along PC2. And then for the 4 donors that were decomposing at cooler temperatures, we just had 2 stages, which was the early and the middle. And these donors never really reached their late stage prior to stopping our trial. And so again, the early stage samples were tightly clustered, while there was variability in the middle stage samples along all of the principal components. Here's just a chromatograms just to show you what the early, middle, and late stage would look like. Comparing these to prior studies, so prior to the current study conducted in Canada, there have been 3 previous studies conducted on human donors decomposing in an outdoor environment. One of which was actually conducted in Texas by the organizers of this workshop. And they reported 58, a list of 58 VOCs in human decomposition. And we detected 32 of those VOCs in our study, of which dimethyl sulfone was one compound that we reported prominent, as well as was among the list of 58 VOCs in the prior study. And the other 2 studies were conducted in Australia. The one study compared the trends at warmer and cooler temperatures. And when we compare that to our study, we found alcohols and aromatics were generally more dominant at warmer temperatures, while there's hydrocarbons, which we called linear aliphatic and cyclic aliphatic, that were more dominant at cooler temperatures. But just to be mindful, warmer and cooler temperatures in Australia and Canada would look a lot different. And the second study conducted in Australia did not report any trends in the class. And they just reported that there was a great deal of variability between individual donors. Now, there can be a lot of future work done, but then I think we can focus on actually doing these studies in different geographical locations just to be able to understand if there is actual variability in the odor profile depending on the locations or not. And Canada is actually characterized by the presence of snow 4 to 5 months a year. So developing a method where we can study the wool-rite profile in snow would be an interesting find, as well as this study actually focused on analyzing soft tissue decomposition, and it was stopped when the donors reached their skeletal stage. And so actually studying dry bones would also add value. And then decomposition in land and water is quite variable. And so developing a method where we can analyze VOCs from donors decomposing in water would also add value. And that brings me towards the end of my presentation. And I'd like to thank all of my professors, everybody that's been a part of this presentation, and all of my funding agencies. Thank you.